Hello and welcome to, you guessed, another very entertaining video. And similar to what we did with the sympathetic routes, I'd like to diagram out the parasympathetic routes to help us understand and be able to compare and contrast some of the differences between these two unique systems. Now you might be asking, why am I staring at a guy's face showing submandibular, sublingual, and parotid salivary glands? Well, let's bring in another picture. While my artistic rendering of a donut might not make you salivate, I want you to close your eyes for a second and think about your favorite donut, or maybe you don't like donuts. Think about your favorite food. Now feel those salivary glands just crunch down as the saliva starts to fill your mouth. What's that? Maybe even a little tear we see dribbling out of your eye as you think about the delicious treat that you wish you had right in front of you right now. That sensation of just thinking about food and then the mouth preparing for food as the salivary glands constricted to release their saliva into the mouth. This is what the parasympathetic system is all about. So often we'll say the parasympathetic, as we've talked about, rest and digest. So now that I've got you craving your favorite food, and we're all ready to start this process, let's look at how these things are wired. As we contrast this with the sympathetic system, let's recall that from the sympathetics, if we were looking into the central nervous system, which is brain and spinal cord, the sympathetic preganglionics all originated in the spinal cord, from the first thoracic vertebrae, thoracic 1, T1, through about L2, L3 sometimes, lumbar 2. That's where the preganglionics originate. The sympathetics is a different story. For the sympathetics, we need to be up in this region and just a little bit down here at the bottom. But as this region of the brainstem is our primary focus, let's zoom in on this. We're going to start at the top of the brainstem with the midbrain, and then transition to the hindbrain, which we're going to break down into the pons here first, and the medulla oblongata beneath it. And what we'll see is that over 90% of the parasympathetics are originating from this region. Less than 10%, maybe even down to 5%. We'll go with 10. It's a nice round number. Down here from the sacral spine. Now, if you're familiar with neuroanatomy, you might be thinking, well, I bet these are cranial nerves, and you're exactly right. For example, parasympathetics to the eye would follow the oculomotor nerve that starts up here in the midbrain. There's the nucleus. This would be cranial nerve 3, the oculomotor nerve. That's going to carry some sympathetics. I'm not saying the whole thing parasympathetics. I'm not saying the whole thing is parasympathetic. I'm saying there are parasympathetic neurons in the cranial nerve. Cranial nerve 5, the trigeminal, will carry parasympathetics to the lacrimal gland to help with crying. Cranial nerve 7, facial nerve, going back up to the salivation that we were experiencing as we thought about that donut. That's going to be part from the facial nerve. Also part from the glossopharyngeal nerve. Now as we get down in here into the medulla, there's cranial nerve 9. And arguably, the most important cranial nerve for parasympathetics will draw emanating down here from the base of the medulla. It's really coming out of the remnants of embryonic branchial arch number 4. This is the vagus nerve, cranial nerve 10. We'll underline it a couple of times because this is important. Why? Because this is going to provide parasympathetics to everything from the heart up high in the thoracic cavity, all the way down to the kidneys, retroperitoneal down in the abdominal cavity. The only thing we haven't touched, if you're paying attention, is the pelvic floor. The organs on the pelvic floor, we're talking about bladder, we're talking about reproductive, and those, as you might have guessed, are that last 10% coming from the sacral spine. So there are the origins of the preganglionics of the parasympathetic system. Now, as we continue to contrast the parasympathetic with the sympathetic, recall that the sympathetic typically was a short preganglionic with a long postganglionic. What might this look like? Well, from the previous video, we remember that we started in the spine, in the lateral horn of the spinal cord, and then there were several routes that we could go. Regardless, the preganglionic was myelinated. We synapse in a ganglia. It could have been the sympathetic chain ganglia. It could have been a collateral ganglia. Postganglionic was unmyelinated, long, and we're off to the target tissue. You should be able to go through and fill out the neurotransmitters and the receptors and everything involved in that. Now let's look at the parasympathetic. Typically just the opposite, a long preganglionic with a short postganglionic. Like the sympathetic system, the preganglionic is myelinated, whereas the postganglionic is not. Now, for this parasympathetic system, let's label quickly the neurotransmitters. This is acetylcholine, acting on N2 nicotinic ionotropic channels. From the postganglionic, it's also acetylcholine, acting on target tissue containing G protein coupled receptors. These are muscarinics, M1 through M5. So it could be M2, M3, M4. There are 5 different types. The odds are stimulatory, the evens are inhibitory, typically, just like we see in the sympathetics. And now we should be able to trace the routes for both systems. If we're specifically looking at sympathetics, let's consider, for example, the heart. In a rest and digest scenario, what do I want parasympathetics to do? I would expect parasympathetic stimulation to decrease heart rate and lead to a less forceful contraction. So if we were to trace the route, we would start the vagus nerve, up in the brainstem, and we're going to follow this particular branch. There's my long preganglionic, and then right at the target tissue, here's my very short postganglionic. Now, importantly, what do we call this ganglion? Because we're synapsing right at the target tissue, there aren't a lot of common names for ganglia. There are some, but we're just going to generalize this and call this a terminal ganglion because it's happening at the end of this long preganglionic. In both cases, this is going to be acetylcholine. In both cases, I'm going to need acetylcholinesterase to go clean out the acetylcholine after we're done. And as we discussed, because this is parasympathetic, we would expect this to lead to a decreased heart rate. And we should feel comfortable creating those traces for every major organ in the body.